Hello everyone, this is Pradeep Kumar and today I will give an overview on an important topic of cytology, that is cell cycle and cell division. All the organisms arises from a single cell by its continuous growth and ability to divide cell into new daughter cells so that it can multiply in its number either for tissue formation or formation of gametes. The cell division activities includes number of repeating phases that forms a cell cycle. So first of all, we will see how the cell divides. Suppose I have to divide this board into two equal half. I will cut from the central margin into two equal halves. But if this board has some component like this, and now I have to cut these components along with this board. How do I do that? I will just bring them on the same margin and now if I will cut from the same margin, I will get these components and the board into two equal halves. Similar is the concept of cell division. What happens there? Let's see. This is a cell having central part called nucleus. And the membrane of nucleus is called nuclear membrane. And the cell is filled with cytoplasm. Around the nucleus there is endoplasmic reticulum. And in the nucleus there is nucleolus. And the nucleus contains the hereditary material called DNA. These DNA become compactly packed and become more condensed to form chromosomes. If there is a single set of chromosomes in a cell, then the condition is called as haploid. And if there is pair of set of chromosomes in the cell, then the condition is called as diploid. So what happens for the cell divisions? This endoplasmic reticulum, nuclear membrane and nucleus start disappearing, due to which the chromosomes become free from the nucleus like this, and after that these chromosomes will arrange on the central line of the cell like this, with the help of spindle fibers that arises from the poles. These are called as spindle fibers. Now after that, the cell get divides along with these chromosomes into two equal halves. In the next phase, the divided chromosomes will start moving towards their poles. After that, when the chromosomes moves towards the poles, the nuclear membrane starts reappearing along with endoplasmic reticulum and nucleolus. So in this way, a single cell divide into two new daughter cells. So overall, we see that the cytoplasm of the cell is dividing and the nucleus is also dividing. The division of nucleus is called as karyokinesis and the division of cytoplasm is called as cytokinesis. Division of nucleus is called as karyokinesis and the division of cytoplasm is called as cytokinesis. So this early stage of the cell division is called as early prophase. And this stage is called as late prophase. In the late prophase, the nuclear membrane disappears along with the nucleus and endoplasmic reticulum and the chromosome becomes free. And in the next stage, the cell division takes place along with the chromosomes. Before that, the chromosomes arrange on the central line. This stage is called as metaphase. And after that, the chromosomes, divided chromosomes, moves toward their pole. This stage is called as anaphase. And when the chromosomes move toward their poles, then the nuclear membrane, endoplasmic reticulum, and nucleus starts reappearing, and the two new daughter cells will form. So this is all about cell division. Now let's see how many types of cell division are there. There are three types of cell division. Let's see. Types of cell division. So there are three types of cell division. Mitosis, meiosis, and amitosis. In mitosis, what happens? The diploid cell divides into two new daughter cells which are also diploid. So in this mitosis, the parent cell is dividing into two new daughter cells which are identical to their parents. And this happens in the formation of tissue. And this division is also called as equational division. Because the number of chromosomes will remain the same in the daughter cell. In the meiosis, the diploid cells divided into two stages. First stage and second stage. So in the meiosis, diploid cell divide into four haploid daughter cells in two stages, first stage and second stage. The first stage is called as reductional division because the diploid cells is dividing and the chromosome number will become reduced to half. And in the next second stage, this is equational division. Because from the first phase to the second phase, the number of chromosomes will remain equal. And this happens in the gamete formation. In the mitosis, a diploid cell can give rise to the diploid cell, and a haploid cell can give rise to the haploid cell. And this happens in only prokaryote cells. While mitosis and meiosis occurs in eukaryotic cells. So this is the all about the types of cell division. Now the cell division have number of phases, and that forms the cell cycle. So let's see the cell cycle. In the cell cycle, there are mainly two phases. First one is interphase, and another one is amphase, or it is also called as divisional phase. And this is also called, interphase is also called as I phase. In the interphase, there is further three phases, that is G1 phase, S phase, and G2 phase. In the G1 phase, cell grows very rapidly, and there is also protein synthesis and RNA synthesis, and enzyme also synthesized in this phase for the DNA replication. Rapidly grows. In the NS phase, DNA replications occurs, and the amount of DNA will get double, but the chromosome number will remain the same. The DNA gets replicates to become double, and in G2 phase there is protein synthesis and RNA synthesis only. In the cells, there are four stages that we have already discussed. That is prophase, metaphase, anaphase, and telophase. So let's see these stages distribution in the cell cycle diagrammatically. This is M phase. And this is completely I phase or interphase. This includes 95% of the cell cycle. Now after the M phase, there is a small phase called G0 phase, in which the cell cycle get arrest and a new cell cycle begins, and after that there is another phase called G1 phase, and then S phase, and then G2 phase, and in the M phase have prophase, metaphase, anaphase, and telophase. After that there is cytokinesis. Cytokinesis in which there is division of cytoplasm. This is prophase, metaphase, anaphase, and telophase. This covers only 5% of the cell cycle. So this is all about the cell cycle. So let's see some questions based on this. During which phase of cell cycle amount of DNA in a cell remains at 4C level if the initial amount of amount is 2C. Option denoted as 2C. Option A, G0 and G1. Option B, G2 and M phase. Option C, only G2. Option D, G1 and S. So what the question says, if the cell cycle initially if the amount of DNA is 2C, and in which phase the amount of DNA will become double, that is 4C. So what we have seen in the cell cycle, there is M phase, a small G0 phase, and after that there is G1 phase, S phase, and G2 phase. So this is G0 phase. In the G0, the cell cycle gets arrested and the new cell cycle begins. So initially the amount of DNA is 2C in G1. Now the cell cycle will enter in S phase and the amount of DNA is 2C. Now this amount of DNA will get doubled till the end of S phase, and when it enters in G2 phase, the amount of DNA will become doubled, that is 4C. And after that it will enter in M phase, that is prophase, metaphase, anaphase, and telophase. In the prophase stage, the amount of DNA will remain the same, that is 4C. And in metaphase also, the amount of DNA will remain the 4C, but from the metaphase stage, the cell will divide and it will distribute into two equal parts. That will appear in anaphase as 2C and 2C. and the telophase also remains 2C and 2C. So the first option is G0 and G1. So G0 is the arresting phase. So this is wrong. And in G1 phase, the amount of DNA is 2C. So this is also wrong. In second option, that is option B, the G2 is 4C. So this is correct. In M phase also, the amount of DNA is 4C, but from the anaphase, it will divide into two equal parts in 2C and 2C. So this can also be correct. Only G2, so this will become wrong. And in G1 phase, there is 2C only. In S phase, the amount of DNA will start become double, but it is not double. So this option will also become wrong. So the correct option is option would be, that is G2 phases and M phase. So let's see another question. In S phase of cell cycle, option A, chromosome number will increase. Option B, amount of DNA remains the same. Option C, amount of DNA will become doubled. Option D, amount of DNA reduced to half. So in the S phase, the amount of DNA will increase, or the amount of DNA will remain the same, or the amount of DNA will become doubled, or it reduced to half. So we have seen in the S phase, the amount of DNA will become double in its volume, but the chromosome number will remain the same. Only the amount of DNA get doubled. So option A will be wrong, option B will be correct, C will be wrong, and D will also be wrong. So now let's see one more question. During mitosis, endoplasmic reticulum nucleus begin to disappear at option A, early prophase, option B, late prophase, option C, early metaphase, and option D, late metaphase. So during mitosis, endoplasmic reticulum and nucleus will disappear. We have seen in the early prophase stage, in the nucleus, the DNA become more condensed, and in the late prophase, these the nuclear membrane, endoplasmic reticulum, and nucleus start disappearing, and after that, there is the chromosome become free in the late prophase stage. This early prophase stage. After that, in the metaphase stage, early metaphase stage, the chromosomes will get arranged and the nuclear divisions takes place along with the cell. So the endoplasmic reticulum, nucleus, and the nuclear membrane start disappearing from the late prophase, so that the chromosome will become free. So the correct option is late prophase. Option A is wrong, option C is wrong, and option D is wrong. The correct option is late prophase. That's all for the day, and have a nice day.